Our journey begins with NCBI, the National Center for Biotechnology Information. Open NCBI.nlm.nih.gov and search for any gene such as TP53. You'll find detailed information including the FASTA DNA sequence. Next, head to blast.ncbi.nlm.nih.gov and choose nucleotide blast. Paste your sequence and hit search. Instantly, you'll see an alignment map showing how your sequence compares to others in the database. This process is called a homology search, and interpreting e-values helps you understand the significance of matches. These tools are fundamental for genetic research, allowing you to quickly identify similarities and differences between sequences. Next, we move from genes to proteins using UniProt and ExPASy. Visit UniProt.org and search for TP53 human. Here you can explore protein functions and domains and download the FASTA protein sequence. To translate DNA into amino acids, jump to ExPASy.org and use the Translate tool. Paste your DNA sequence and instantly see the corresponding amino acid sequence. This workflow bridges the gap between gene information and protein function, helping you understand how genetic variations impact biological processes. UniProt and ExPASy are invaluable for researchers studying protein structure, function, and evolution. Visualize genomic context in regulatory regions. Ensembl and UCSC Genome Browser are your go-to tools. Start at Ensembl.org and search for BRCA1. You can explore transcript variants and click location for a detailed exon view. Then switch to genome.ucsc.edu, paste the same gene ID and toggle features like CPG islands and conservation tracks. These platforms allow you to see how genes are organized, regulated, and conserved across species. Understanding genomic context is crucial for interpreting gene function, regulatory elements, and evolutionary conservation. Protein structure analysis, PDB, and AlphaFold database offer powerful visualization tools. On RCSB.org, search for protein IDs like 1TUP or BRCA11JM7. You can rotate the 3D protein model in the viewer, examining its shape and folding. Next, visit AlphaFold, ExPASy, and type the same protein to compare predicted structures with experimental data. These resources help you inspect protein folding and assess model confidence, which is essential for understanding function and designing experiments. PDB and AlphaFold are revolutionizing structural biology by making protein models accessible to everyone. Explore transcriptomic data. GEO and ArrayExpress are essential platforms. Open NCBI.nlm.nih.gov/geo and search for datasets like breast cancer RNA-seq. Download a series matrix file and view expression levels in Excel. Then visit ExPASy.uk/ArrayExpress and search for similar datasets. These tools allow you to locate and preview transcriptomic data, helping you analyze gene expression patterns across conditions and experiment. GEO and ArrayExpress are widely used for large-scale studies, enabling researchers to access and interpret high-throughput sequencing data. Homology search is a fundamental concept in bioinformatics. It involves comparing DNA or protein sequences to identify similarities that may indicate evolutionary relationships or functional conservation. Tools like BLAST make this process accessible, allowing researchers to quickly find matches and interpret e-values, which measure the significance of the alignment. Understanding homology is crucial for gene annotation, functional prediction, and evolutionary study. By mastering homology search, you can uncover hidden connections between genes and proteins across different organisms. The process of translating a gene's DNA sequence into a protein's amino acid sequence is central to molecular biology. Tools like ExPASy's Translate tool automate this conversion, helping you understand how genetic information is expressed as functional proteins. This concept is vital for studying mutations, protein function, and disease mechanisms. By moving from gene to protein, researchers can link genetic variations to biological outcomes, paving the way for targeted therapies and personalized medicine.